So here quickly is an overview of cell respiration, which just shows the mitochondria and the location where each process occurs. So to start out, in the cytoplasm, glycolysis occurs. Glycolysis is a metabolic pathway that converts sugar into pyruvate. So as we can see here, it starts out with glucose and goes through a couple of intermediates, all 6 carbons, and actually uses 2 ATPs, which releases ADP. It actually uses 2 ATPs and is phosphorylated. And then once it's a fructose 1,6 biphosphate, which is also 6 carbon, it goes through lysis, which again goes through several intermediates. And here, after the initial lysis, there's actually an oxidation which produces the NADH, which is actually what's going to be released to the electron transport chain. And then through the rest of this, the ATP is formed, and that's through several intermediate units as well. And then once the ATP is produced, it also produces 2-pyruvate. So then glycolysis gives this ATP and the pyruvate to the link reaction, which is essentially what links glycolysis and the Krebs cycle together. So in the link reaction, it takes a pyruvate, which was produced in glycolysis, and then is going to end up with acetyl coenzyme A. So an NAD, which is not reduced, is reduced here with an H from the pyruvate to form reduced NAD. And then a CO2 is released as a byproduct, which is a decarbolization. And then coenzyme A is introduced, and that forms the acetyl coenzyme A. And then here, this reduced NAD is going to also be released to the electron transport chain, and then the acetyl coenzyme A is what's going to become the first step in glycolysis. In the Krebs cycle. So for the Krebs cycle, it starts out with coenzyme A and goes through several different processes. As you can see, it's a very complex circle. The main points here are that for every turn of the Krebs cycle, there's going to be 3 reductions of NAD, which are shown here, here, and here. 1 reduction of FAD, which is shown here. 2 decarboxylizations, which are here and here. And then it's going to produce 1 molecule of ADP, again through phosphorylation. And then this is also known as the citric acid cycle, because once you remove the coenzyme A, you get citric acid. And then again, all of these reduced electron carriers are going to go to the electron transport chain. So that's the reduced FAD and the reduced NAD. So the final step, and the step that produces the most ATP, is the electron transport chain, which is also known as the ETC. So here it takes place in the intermembrane space, and this is the intermembrane up here, and this is the inner mitochondrial membrane, and then this is the matrix. So to start out, all of the electron transporters that were given to the ETC are oxidized, so they lose their electrons, and then the protons go through to the inner membrane. So that's this process here, which is the oxidation of each of these. And then this is going to release H plus protons into the intermembrane, which is going to build up a high concentration of H plus, which is we're going to need for ATP synthase, while producing a low H plus concentration in the matrix. And then these electrons that are given off by the oxidation of the electron carriers is going to travel through this path here, as shown in the red, and then it's going to go together with O2, which is a terminal electron acceptor, and additionally with 2 hydrogen protons, it's going to form water. And then finally, once all of this is completed, there's enough H plus or protons in the inner membrane to activate ATP synthase, which is going to take ADP and phosphorylate it to produce a lot of ATP. So overall, that's cell respiration.